Okay, welcome back, students. So today, an important lecture, we are going to talk about transcription. So one of the three big important lectures for the final exam. So just going to let a couple people in here. I think that's everybody now. Okay, so last week we talked about replication. This week we're going to talk about transcription. So transcription is the process of making RNA using a DNA template. So you can see actually in this diagram right here, we have some transcription going on. And we have two strands of DNA and we're making one strand of RNA from this. And the enzyme that does this whole process is called RNA polymerase. So let's talk a little bit about transcription and then we'll talk about how it works. So one thing to remember is that we're going to make RNA. So RNA is a little bit different than DNA. Generally, RNA is single-stranded. It's not always the case. I think I showed you with the transfer RNA that sometimes it ends up double-stranded regions, but it's not consistently double-stranded like DNA. And if we look at the chemistry of RNA, one of the big things is the R. So the R stands for ribose, and the D for DNA stands for deoxyribose. So if you take a look, it has a sugar in it, and the sugar is almost identical. The only difference here is at the 2' position. In DNA, we have just a hydrogen attached instead of an OH. So it's a deoxyribose. So technically, it's a 2' deoxyribose. As you can see, I have two. Usually there should be a little prime in there. But usually just for short, we call it deoxyribose, and that's where DNA gets its name from. So tiny difference, but it's enough that enzymes that recognize RNA and DNA can actually tell the difference, and because you've got a polar oxygen group there. The other thing that's different, and you probably know this as well, is that in RNA, we use a different nitrogenous base. We don't use thymine, we use uracil. Very similar in structure as well. You can see the differences that thymine has an extra methyl group. So same thing, it helps enzymes recognize DNA versus RNA or the other way around. So slightly different nucleotide. So there's some RNA right there, and single strand, you can see there's the 4 nitrogenous bases. It's also made out of phosphates. So one nucleotide is going to have a phosphate. It's going to have pentose sugar and a nitrogenous base. So that would be one RNA nucleotide. RNA nucleotide. So just a reminder, I am going to final exam, going to have a big question about the 6 types of RNA. Haven't decided the question yet. Usually, I think I told you before, usually what I do is have it's a 6-mark question, and it will have the type of RNA on one side and the description or definition on the other side, and it's kind of fill in the blank. So you fill in the definition, description, or you fill in the name. And so don't be surprised if you see that exact question. So when we, last day, we talked about a couple of different types of RNA, talked about the telomerase RNA, and today we're going to talk about messenger RNA, and we're going to talk about small nuclear RNA. So let's talk about transcription. In transcription, we're making RNA. And the process, excuse me, a little frog in my throat today. Process is a little bit different from replication because we're not making DNA, we're making RNA. Although we are going to use DNA as the template. The sequence that's found in the DNA template strand, in fact, is going to be used to determine the RNA sequence. Don't forget, we're going to use uracil instead of thymine, so U's instead of T's. And the enzyme is called RNA polymerase, or you can call it RNAP for short. So there's RNA polymerase. There are different types of RNA polymerases, and I'll think about a slide discussing that in a minute. If you are a bacteria, though, you just have one type of RNA polymerase. If you're a eukaryote, I think you have 3. Unless you're a plant, I think you've got 4 or 5 types. I'm not sure why plants have extra types. But you can see there's the DNA. You've got the single strand of RNA being made, and you've got the enzyme zipping along. So one thing about RNA polymerase that you should know is that RNA polymerase does not need all those accessory enzymes like DNA polymerase. Remember, DNA polymerase needed a helicase, and it needed the topoisomerase, and it needed the single-stranded binding protein, all those kind of things. So it turns out RNA polymerase can do all of that stuff. It can unzip, it can synthesize, it doesn't need a primer, and so it's a little more versatile than DNA polymerase. So why is that? Well, we don't need to be as accurate when we're making RNA. If you make a few mistakes, it doesn't matter. You can always make more RNA. You're not affecting your entire genome for several generations to come. Whereas DNA polymerase has to be extra careful. The proofreading is a lot better. RNA polymerase does have proofreading, but it's not as good as the DNA polymerase proofreading. I told you I had a slide. So like I said, if you're a prokaryote, you have one type. If you're eukaryote, you have 3 types, unless you're a plant. For some reason, plants seem to have extra types. So you can see in eukaryotes, different RNA polymerases are transcribing different types of RNAs. So RNA polymerase 2 is the important one. And this is kind of the one we're going to be talking about today because it transcribes messenger RNA. And messenger RNA, of course, is going to transcribe RNA that is going to use to go to the ribosome for translation to make proteins. So that's kind of the one we're thinking about. Okay, so some very important stuff here. Usually I draw this on the board, and but I want to talk about what is going on with some of this 5' stuff and this 3' stuff, and talk about the different strands of DNA. So if you take a look, I have a strand of DNA. And of course, there's 2 strands because it's a double-stranded molecule. And one of the strands, you can see this one here, is going 5' to 3'. And the other one is, of course, anti-parallel. So remember, anti-parallel means the one strand is going opposite direction in terms of how the chemistry is organized. So 2 strands each have a name. So one strand we call the sense strand. So the sense strand, you can see my note there, it says it's the same sequence as the messenger RNA. So if our messenger RNA, or sorry, if our DNA was, let's say, our start codon is ATG, that would mean that my messenger RNA be AUG. So it's not perfectly identical because all our T's are substituted and we make them into U's, but for all intents and purposes, the sense strand is the same as messenger RNA. So that would make the other strand the antisense strand. I don't like that term as much, antisense. It's used pretty common in the literature and in textbooks. A better term that I like to use is the template strand. So this is the strand that the RNA polymerase, the enzyme, is actually looking at. This is the one it's binding to, and it's providing complementary RNA nucleotides. So sense, antisense, template, you should know all of those words. Hopefully it's not lots of, hopefully it makes lots of sense, but 2 strands and we have to start somewhere. The other one note to say about the sense strand is that this is where the start codon is. This is where the start codon. So ATG, remember, is the start codon for DNA. So my messenger RNA, like I said, it's going to be look a lot like the sense strand. It's not going to be the full genome. Your messenger RNA is just a little chunk of the genome. It's usually 1 gene or sometimes a couple of genes, as we'll find out later. And then that messenger RNA is going to find a ribosome eventually. So this is something I want you to keep in mind when we talk about the details of transcription. So usually when we make our messenger RNA, we're going to have a fragment of DNA where the gene is. So our gene, you can see, is there is one section of DNA. And we talk about any nucleotides before that are upstream. And any nucleotides afterwards are what we call downstream. So you notice that my messenger RNA it takes the gene and it takes a little bit before and a little bit after. It's trying to just to make sure, you know, we want to make sure we get the whole gene. So sometimes you have to be careful and just get everything. And that's kind of part of what is going on. There's also some regulatory stuff that's going on before and after, upstream and downstream. So if you ever hear, if you take some more future molecular biology classes and they talk about upstream of the gene, it means before the gene. If they say downstream, it means somewhere afterwards. Or you might talk about gene number 5 is downstream from gene number 2 or something like that. So these regions, these regulatory regions, the first one to mention is something called the promoter. And so I'm going to write a definition for promoter. Hold on a second. Let me just, I'm going to write a definition for promoter, and I'll come back to that second one in a moment. So what is a promoter? This is an important word to know for Biology 107 final exam. So the promoter is a sequence of DNA. And you can maybe see that. I have that little box on my DNA strand. Sequence of DNA. And what happens at that sequence of DNA? And this is where the RNA polymerase binds. This is where the RNA polymerase binds. And this is, of course, where it starts transcription. So that's what a promoter is. RNA polymerase needs to know where to start. It doesn't start at the start codon, it starts before the start codon. And like I said, this is for regulatory purposes. Plus, if you started at the start codon, you know, you could miss something. You might miss the A and just start off with TG or something like that. So sometimes, like when we make things, you know, in person, sometimes you make a little extra. In this case, we're making a little extra RNA. So the promoter itself is upstream of the gene, not very far, 5, 15 nucleotides. It's not really very far at all. And so it's just important, like I said, to make sure you get the entire transcript, you get the entire gene. You don't want to miss anything. The promoter also determines which strand is the template versus the sense strand, because it's looking at and finding the promoter, and that helps the RNA polymerase figure out which strand to use. But I'm not going to include that detail for this class. So downstream is another thing there, and this other thing is called the terminator. So what is the terminator? It's very similar to the promoter in that it's a sequence of DNA. So terminator is a sequence of DNA. It's found downstream of the gene. Maybe I forgot to put that with promoter. You could say it's a sequence of DNA before the gene where RNA polymerase binds. So that's something that, you know, you could include that if you like. A sequence of DNA after the gene. And this is basically where RNA polymerase stops and detaches. So where RNA polymerase detaches and stops transcription. Detaches and stops transcription. Sorry, I had to cram that in there. I got to think about planning these PowerPoints around making sure I have space for the notes in there. Okay, so promoter, terminator, sense, template. You know, we're going to start throwing these words around. Okay, so if you're not comfortable with them today, make sure you go learn them, because they're going to come up again in the next few lectures, and they're going to be important words to know for the final exam. You need to know what a promoter is for the final exam. You need to know what a promoter is for topic 18. So make sure you do know these things. So let's talk about the details of transcription. It's really not as complex as replication. I'm not going to draw you a big fancy whiteboard thing. We're going to break it down to 3 stages. Initiation, that means start. Elongation, that means growing. And termination, that means stop. So you can see there's the RNA polymerase, and you can see it's binding the DNA and adding in those complementary nucleotides. So step 1 of transcription is basically that RNA polymerase, it binds the promoter. So I've already told you That's what the promoter does, and the promoter says, hey, start here, and it's a little bit before the start codon. And then it basically starts doing its thing. It unwinds the DNA, and it starts adding in those complementary nucleotides. And remember that all DNA and RNA is always 5' to 3'. There it is right there, 5'. My pen is not working suddenly. Let me try this again. There we go. Sorry, I don't know what's going on with my pen. Crazy stuff. Mondays. Always 5' to 3'. Okay. So remember that. Remember, that's how those enzymes work. They work at the 3' end. So you're going to see as this little RNA transcript comes off, you're going to see how that works. So elongation is basically it's moving along, and it's making several complementary nucleotides at the same time. So it's reading the template strand, remember, and it's matching those complementary base pairs. So if it sees an A on the template strand, it's going to add a U. If it sees a C, it's going to add a G. If it sees a G, it's going to add a C. If it sees a T, it's going to add an A, and so on. And notice it's making this transcript in the 5' to 3' direction because the enzyme has to use that 3' end. It can't add on to the other end. That's the way the enzyme works. And so the DNA basically rewinds, and those hydrogen bonds reform behind, and the DNA remains intact this entire, basically after the process is done. Last step is termination. So after the gene is that terminator sequence, and the terminator sequence says, okay, this is the end, and the enzyme detaches, and the RNA goes its way, the enzyme goes its way, and they do their things. The enzyme will get reused and make more RNA, and the RNA itself will probably find a ribosome. It's not going to find a ribosome right away. You're probably thinking, why is that? Well, it turns out that ribosomes are, of course, out in the cytoplasm. So at least in a eukaryotic cell, it wouldn't find a ribosome until it made it out of a nuclear pore. So I have a little video here for you. It's pretty short, but it uses some of the same images from the textbook. It's about prokaryotic transcription. So this is bacteria. You may notice the big issue here. I don't really know why they do this, but that is a nucleus. There should not be a nucleus for a prokaryote, but the video itself is fine other than this nucleus part. So whoever made it way back in 2001, bad on them, but there are lots of videos out there. I like this one because it's short and sweet, and it looks a lot like the textbook images. And other than that, maybe someday I'll find a better one, but there's lots of really long ones out there. And it's really not a complicated process. So I'll press play for you.
The first step in protein synthesis is transcription of RNA from DNA. The transcription of RNA from DNA begins with initiation. An enzyme called RNA polymerase attaches to a region of the DNA called the promoter, which lies next to the beginning of a gene. RNA polymerase pries the DNA strands apart, and RNA nucleotides line up along only one of the DNA strands, the template strand. RNA polymerase links the nucleotides together. The second stage of transcription is elongation. RNA polymerase moves along the DNA, adding nucleotides to the end of the growing RNA molecule. The RNA strand peels away, allowing the two separated DNA strands to come back together. Here's a close-up view of elongation. RNA nucleotides pair up with their complementary DNA bases on the template strand. Note that U in RNA pairs with A in DNA. RNA polymerase moves along the DNA strand, linking nucleotides to the end of the RNA strand. The last stage of transcription is termination. Once RNA polymerase has gone past a sequence called the terminator, the enzyme releases the completed RNA and detaches from the DNA.
Okay, so hopefully pretty straightforward. Like I said before, make sure that these processes, replication, transcription, and translation, that you can comfortably describe them for the final exam. This is a pretty typical kind of long answer question, is to describe one of these processes. And start here. This is the easiest one. So when you're studying, this is a good place to start. So I want to talk a little bit about eukaryotic transcription because like everything eukaryotic, it's more complex. First thing to note is most of what we just saw was exactly the same. You still have 2 strands of DNA. You still have an enzyme called RNA polymerase. This one's just called RNA polymerase 2 because eukaryotes have more than one of them. One thing that's interesting is that a lot of eukaryotic organisms have this common promoter. It's called a TATA box. So you can see that that's exactly just described in the sequence TATA. And if you read the sequence on both strands, it's TATA on both strands. So why a TATA box? Well, it turns out that T and A have only 2 hydrogen bonds compared to C and G. So it probably is just a little bit weaker and in terms of the amount of hydrogen bonds. And so it's easier to separate the strands there. But other than that, it has a promoter and it's going to have a start codon in there somewhere, where the gene is. So the gene probably starts here somewhere. We're going to have a template strand and a sense strand, just like before. So one new thing to know about eukaryotes is we use something called transcription factors. So what is a transcription factor? Why don't we just say transcription proteins? Because turns out some of these things aren't just proteins. They might have RNA parts, but mostly they're proteins. And these are kind of specialized regulatory proteins, and they have to bind before the enzyme RNA polymerase can bind. So once they bind, I'll come back to those transcription factors in a second. They form this thing called a transcription initiation complex. So that's the RNA polymerase plus the transcription factors, and then this thing can start. So why do we need transcription factors? You're probably wondering, what are these things all about? Well, think about it. We are complex organisms, and they are mostly regulatory, in fact, in nature. We have so many different cell types. We have red blood cells, we have white blood cells, we have neurons, we have skin cells, we have liver cells, we have bone cells. And so we need to tell our body which genes to turn on in different tissues. We also grow, and we start off as juveniles, and there's different things going on when you're a fetus, different things going on when you're a baby versus a toddler versus a child or a teenager or an adult. And so we need to be able to have a way to turn on or turn off genes. And that's what these transcription factors are all about, is they're just basically regulatory components, and they're required for the RNA polymerase to do its thing. So just 2 extra really added things about eukaryotic transcription is to at least get started. One is the TATA box, 2 are the transcription factors. And of course, another term to learn, which is transcription initiation complex. So there is a transcription factor, and you can see that this one here is interacting with the DNA. It has these cool DNA binding domains. They kind of fit into the groove of the double helix. They're kind of cool enzymes. And there's a little bit more of a complex model of regulation in eukaryotes. I'm not going to go into all of this, but you can see it can get really complicated. There are other DNA components called enhancers. Sometimes there's silencers and activators and repressors and all of these things. But a lot of these things here, like A, B, and so on, all of those are transcription factors. And there's our RNA polymerase, and it looks like it's ready to go. So back to this diagram here. We have our sense strand and our template strand, promoter, our terminator, and a couple other things to say about eukaryotic RNA. So notice at this point, I'm not calling it messenger RNA yet. I'm actually calling it pre-messenger RNA, or an unprocessed transcript. Sometimes you hear people call it. It requires a little processing, it turns out, in eukaryotes. Why? Because we're more complex. We have a nucleus, and we have other things going on. So this whole process is, of course, a little bit more complex. So one thing you can see, it says here that often at the end of an RNA sequence is this thing called a polyadenylation sequence, which is usually mostly a bunch of A's. And the reason for that is it just helps the whole thing to be a little bit more stable. RNA is a relatively fragile chemical, breaks down very easily. If you add a little bit of extra on the end, it keeps the whole thing from degrading really quickly. So I'm going to play this video for you. This one's a little bit different from the last one. It is a bit more kind of like animated, and we're trying to be a little bit more accurate around the structures and everything. And it's also relatively short, and they're going to talk about eukaryotic transcription. So I'll play this for you. It's a pretty cool video.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
So that's the end of the video. So I don't know if she mentioned that, but that video apparently is in real time. And so you can see it's a pretty fast process. And I thought they did a pretty good job of explaining what was going on there. So before we get any further here, I wanted to do one of these sample questions. I had mentioned that there's going to be a question like this on the final exam, where you're going to get a strand of DNA, and you're going to have to answer some information about the strand. It's going to be anywhere from, I don't know, 5 to 10 marks. I haven't decided exactly on everything on the final exam. I have to see what I did the last couple of semesters. So what you have enough here now to do, I don't know, maybe 3 to 4 of the marks. So if you take a look here, here's a strand of DNA, and you can see it's actually asking you a couple of things about it. It says identify the sense strand and the template strand. So I'm not making it as easy as I did last time. Last time I made it pretty easy. This time I've made it a little trickier. And so the first thing that you always need to do is look for the start codon. So the start codon is right here. So ATG. If you don't remember what the start codon is, you can use your genetic code, and you can see there's AUG on the RNA, which means ATG in the DNA. So this here, by the way, is not the start codon. That is not ATG. That is 3' ATG. So that means it's 5' GTA. Remember, we're always looking for reading things 5' to 3'. So once you have that figured out, we know where the sense of the template strand is. The sense strand always has the start codon, and the template strand is always the other strand. So there we go. There's 1 mark out of 5 or 8 or whatever I make the question of. Okay, so just labeling the 2 strands correctly. The next, it says give the RNA sequence. So remember that the RNA is going to look basically exactly like the sense strand. So what you need to do is basically copy out the sense strand, and you're going to change all your T's to U's. So notice I'm not starting at the start codon. It actually, the transcription starts before the gene. I'm not exactly sure how far before the gene, so we're just going to copy out everything that you see there. So you can see my TAC becomes a UAC, TAT becomes UAU, and so on until you get to the very end. Notice I did throw a TATA box in there. It's probably too close to the gene. It's usually supposed to be about 15 base pairs away, but I didn't want to make this sequence extra long and extra complicated. It's long enough as it is. So next question C, it says, where are the promoter and terminator sequences? So remember that the promoter is before the gene. So somewhere in there, label a region before the gene, maybe circle the TATA box to make sure that you definitely have the promoter, but it's basically the region before the gene. The terminator is after the gene. So we got to remember our stop codons. And so you can see what I'm going to do is just underline each of the triplets here until I get it. And I'm going to go all the way to the end. And I can see that TAA, that is actually my stop codon right there. UAA, if you can see on the genetic code, is the stop codon. So that means that the terminator is somewhere there after the stop codon. Okay, so that's, let's see here, 1, 2, 3, that's it. Yeah, it's about 3 marks already. And last one is give the amino acid sequence. So I'm just going to underline my codons here. And my protein sequence, I start with AUG, which is methionine. That's my start codon. And UAU, I'm looking up here, it's tyrosine. CCU is proline. And UAA, that's my stop codon. So that's actually it. So like I said, that's about 3 to 4 marks of this question. And still a few other things to talk about. We haven't talked about translation yet, of course, so there's more information to talk about. We'll also want to talk a little bit about mutations and a few other things as well. So I'm not quite done with transcription, by the way. I mentioned that there's some processing that's going to happen in your eukaryotes. So that's what I want to talk about now. So by the way, I think I mentioned before, there is a practice question like this in the, I put a study guide up on Moodle. So if you want to take a look at that anytime you can, and always take a look back at the PowerPoints to see that you've done this right. Because I don't have these answers in the PowerPoint. I just have the question. So if you want, I recommend that you do the question and then go back to the video recording and see that you got it correct. Okay, so let's talk about this modification and this processing that happens here. So that red part in the middle, that is the messenger RNA. And notice one of the first things that happens in the nucleus is these things are added at the end. A cap and a tail, they're called. So 5' cap is at the 5' end, and the 3' tail, or the poly A tail, is at the other end. So poly A, meaning it's lots of A's. So both of these things are they're protective. They're, like I said, RNA is a fragile molecule and it likes to degrade from the ends. So both of these help the RNA to not degrade as quickly from the ends. So that's the first thing to know, is that they're important to protect from degradation. The other thing to know is that they're also kind of like handles. So you can see my second thing here, it says it helps with ribosome binding. So ribosomes, they're going to grab the RNA, and just having a little extra hold on to is going to help it. There are actual ribosome binding sequences that are sometimes found in the promoter regions, but we're not going to worry about those as well. These are just sort of assisting in that category. But number 1 is degradation. And the last thing to know for the 5' cap is it signals nuclear export. I think I actually have a picture of the 5' cap here. Here's what it looks like. And so what does this mean, nuclear export? Well, remember we have those nuclear pores, and things just don't diffuse in and out. They're not free ports. Those nuclear pores have rules of engagement, and that means they let some things out and they let some things in. And so the 5' cap is a signal that says this RNA needs to get out of the nucleus and into the cytoplasm. So the 5' cap is really important for that, because if it doesn't get out into the cytoplasm, the messenger RNA will never find a ribosome, because that's where all the ribosomes are found. You can see this is the structure of the 5' cap. It has 3 phosphates. It has a ribose sugar, and has a guanine. So basically it's a nucleotide on the end, and it's got a little extra methyl group. I'm not sure what the function of that methyl group is. Probably has something to do with recognizing receptors or enzymes or something like that. So here's our RNA, and that's the pre-RNA. It's still called pre-messenger RNA. We're not done processing it. We've added a cap, and we've added a tail. And the last thing that happens in eukaryotes is something called splicing. So notice what just happened is we have these little regions here. We have these things called exons and introns. So what are exons and introns? They are coding, they're sequences of the RNA, and the exons are expressed. So that means they're coding regions. These are the things that eventually are going to be read at the ribosome and become an amino acid sequence. Introns, int is for interruption, meaning they're in the middle of the coding regions. So they have to get cut out. And the word we use for cutting out is this, splicing. So if you ever heard the word splicing, that's what this means. It doesn't mean some scientist in a lab messing with DNA. This is actually a natural process, splicing, where the introns are cut out. So I want to go through this process a little bit and then talk a little bit about why we might do splicing. Because if you take a look at this, this just seems ridiculous. Why are we wasting all of that RNA? And it turns out that we do have some answers, maybe not all the answers for this process. But let's take a little bit look at this splicing process here. So splicing is done by this thing called a spliceosome. And a spliceosome is a big complex, and it's a complex that is made of proteins and RNA. So there is another type of RNA that we're talking about. So today we've been talking about messenger RNA and small nuclear RNA. And like I said, I'll come back and talk about the RNA again and again over the next couple of weeks here. So if you take a look, we've got our spliceosome has something called small nuclear RNA. Maybe I'll mention what that is all about. Small nuclear. This RNA was discovered in the nucleus, and it was small. And so that's how it got its name. It's small nuclear RNA. And eventually we figured out what it was doing. It was part of this thing called the spliceosome, but that's where the name came from. So the spliceosome is this complex of RNAs and proteins, and it recognizes introns, and then it cuts them out. It's like a big enzyme. And there is our cutout intron, and there are exons kind of glued back together. So the small nuclear RNA, by the way, is involved in recognition of sequences. As you can imagine, it's nucleotide. It's also involved in the catalysis. It's also part of the active site of the enzyme. So a couple of things about intron splicing, just sort of footnotes, is that in most cases, RNA splicing is done by these things here, these spliceosomes. Some introns, believe it or not, are self-splicing because they are ribozymes. It's kind of worth mentioning ribozymes as kind of a biological phenomenon, because you'll probably hear about them again if you take more biology classes. So what is a ribosome? A ribosome is an enzyme, and it's also RNA. So this is really weird, because it used to be thought that all enzymes were made out of proteins. Well, it turns out that's not true, that there are some enzymes made of RNA molecules. So this is pretty cool. It's a very small percentage of enzymes. Most enzymes are made out of amino acid sequences. But there are a few out there that are catalytic RNA molecules. So pretty cool. So it turns out RNA can fold into these weird three-dimensional structures sometimes if the sequence is right, and actually form an enzyme. I found this one on the internet. I have no idea what it is. The hammerhead ribosome. I thought that was kind of a cool name. But in this class, we are going to actually encounter two types of ribozymes. First of all, some are involved in splicing. And second of all, some ribozymes are, the ribosome actually has a ribozyme active site. So try not to get that mixed up, what a ribozyme is versus a ribosome. We're going to talk about ribosomes next day. So let's talk a little bit about this splicing and what is going on here. Like, why are we cutting out these introns? Like, what is going on? So generally, the way it works is that exons kind of code for a part of a protein, and it's sort of a self-folding part called a domain. And so it turns out that that there's a lot of genes are what's called alternatively spliced. So if you take a look at this gene here, this is an enzyme called troponin. I'm not exactly sure what it does, but so it makes its messenger RNA. And sometimes you cut out a little bit of this, and sometimes you cut out a little of that. And so you can see in this case here, the one on the left, we've made a messenger RNA out of exon 1, 2, 3, and 5. And on the right, we've made a messenger RNA out of exons number 1, 2, 4, and 5. So these go find a ribosome, and we end up with 2 different proteins. So this is really incredible because now we have a way to use one gene for different gene products. It's kind of wild, really, if you think about it, that we can actually do this. So we have 20,000 genes in the human genome. Doesn't mean we have 20,000 gene products. We actually have more than that, quite a few more. This isn't necessarily applicable with every gene. There's still a lot of questions around splicing, whether, maybe it has something to do with our evolutionary past, or maybe it has something to do with viruses that have infected us. Maybe there are regulatory components. There's still a lot of questions, much we don't really fully understand. We just know it's a very well documented phenomenon. It happens on most of our genes in the human body, which is kind of crazy. In fact, it seems that there are more introns than exons in some genes, which is just kind of mind-blowing, really. There's another place where this alternative splicing is really, really useful, and this is as part of your immune system. So our immune system has these proteins called antibodies, and I don't want to go into antibodies too much, but you probably know that antibodies bind bad things. So they bind viruses and other things, and they have this this pocket on these proteins that binds them. And there are more than 20,000 different potential pathogens out there. So our immune system uses these antibodies to recognize things. So let's say you get vaccinated against COVID-19 or chickenpox or something, and then we make the antibodies, and the antibodies recognize the virus or whatever pathogen it is, and then our white blood cells are going to come along and kill these things. So antibodies are made by alternative splicing, and you have one gene, and it can make many different gene products. So we can recognize many different potential pathogens. So this could be an entire lecture on itself. I'm not going to say anything more about this. So I do have a couple of other things to say, and then I have a Kahoot for you. This is kind of just comparing prokaryotes and eukaryotes. So notice again this thing about transcription factors. I told you bacteria don't have them, but it turns out that archaea in many cases do have transcription factors, which just again makes them a little bit more like eukaryotes than bacteria. Eukaryotes have the splicing and the capping and tailing. Sometimes that happens a little bit in archaea, but not quite so much. So they're kind of a really weird type of organism. Oh, I guess that's it. I thought I had one other thing to say, but this is it. We're going to do a Kahoot. So you can load up the app, and some questions for you. And this one will be medium hard, I will call it. Some of the questions are super easy, and some of them are a little bit on the hard end. So please join in, and we'll play, and we'll talk about the answers as we go along. All right, so let's get started here. Question 1, transcription is the production of. Excellent, everyone got that one correct. RNA, good job. Believe it or not, sometimes people make it to the final exam and they don't remember which one is transcription or translation. Number 2, DNA nucleotides and RNA nucleotides can both contain So the correct answer is phosphate. So remember that thymine is only found in DNA. Question 3. In eukaryotic cells, transcription occurs in the... Okay, correct answer is nucleus. So good job. Okay, I think they're going to get just a little bit harder here once we get to the halfway point. This enzyme is required for transcription. So correct, we're making RNA, so the enzyme is RNA polymerase. Which is not true of RNA polymerase. Okay, so a little bit more of a thinker, but looks like most people got that one correct. So remember, it's always 5' to 3'. So this is backwards. 3' to 5' is backwards. That's why that one's not true. What is the function of messenger RNA? Okay, that one is a little harder, isn't it? RNA carries information. It doesn't actually do anything. It doesn't synthesize, it doesn't translate. So it carries information, and it's just information that's temporary. So DNA carries information between generations, but RNA does not. What would be the messenger RNA complementary to this DNA strand? Okay, I was wondering if I get people with that one. So remember that I can't draw on the screen, unfortunately, but take a look at that DNA. It's 5' TAGGC. So the complementary RNA is going to be it's going to be 3' AUCCG, right? Or if I read it in the correct way, it's 5' GCCUA. So think about that one. Okay, come back and do this Kahoot if you need to, but that is the correct answer. Unfortunately, I don't have any way to draw on the screen here and kind of go through this in a bit more detail, but we'll get some practice with those DNA sequence questions in the next couple of lectures. I'll do at least 1 or 2 more of them. Transcription factors. What do they do? Okay, good job. They're required for RNA polymerase binding in eukaryotes. So that's what they do. All right, so a couple more questions. Which of the following is not a function of the 5' cap? So the correct answer is the 5' cap does not do any proofreading. That is the job of the enzyme. The 5' cap is all about stability and nuclear export, and also helps with ribosome binding. Okay, well, one more question. Mark coming in hot. Non-coding regions of eukaryotic genes are called. Okay, everybody got that one correct. So well done. So maybe that one was easier than I thought it was. Looks like Levi in bronze with 8 out of 10. Michelle a little bit faster to the draw with 8 out of 10. And I'm guessing this is Nina with 9 out of 10. Well done, well done. All right, so that brings us to the end today. So thank you for coming out. Can't stress enough, go home and learn about transcription because you are definitely 100% going to see it on the final exam, and it's going to be worth a bunch of marks. So I'll see you on Wednesday, and you have a great day. Take care.